Hello? Hello, everyone. In a few minutes, we are going to start our today's session. That is part two of CVS Physiology. Let me just perform a mic check. I think I'm audible. I'm going to wait for 2 or 3 more minutes and then we can start. In previous session, we were discussing about the cardiac output and we stopped in the midst of it. हमने basically venous return से पहले पहले वाला portion, that is the intrinsic control of the heart rate, we've already discussed that. ठीक है? We even discussed about some of the different types of pulse do we have, what is the pulse in general. We discussed about the electrophysiology of the heart, leaving out the portions that is will be covered in future ECG sections, which we're going to upload shortly, थोड़े दिनों में. But rest of it all, whether it be the basic properties of your cardiac muscle, or it be the electrophysiology of the heart, or it be the different components which affect your heart rate, we've already discussed that. So today's session is all going to be about mainly your venous return. What are the factors which are going to influence your venous return? And then we are going to move on to discussing something about the circulation as well. Okay? So today I have a lot to be done, and basically circulation in many modules and in different apps that we use. Circulation is considered as a different topic. It is not included in CVS physiology, but we shall be going over the main two circulation that we need to study, at least three circulation that we need to study. That is your coronary circulation and your CNS circulation, okay, and cerebral circulation, I would say. And the third one is your skin circulation, okay. Remaining, that is your placental and fetal circulation. I know that is not basically a part of physiology, and that will be covered mainly in your obstetrics, okay. So let me just wait for 1 or 2 more minutes and then we're going to start at 10:05. Hello everyone, and now we're going to commence our session. Okay, without further ado. So previously we were discussing about the cardiac output. So let me just continue the discussion and discuss with you something about your venous return. So what is a venous return that we have? That is the amount of blood which is returned to the heart via the great veins. That is you're directly related to your stroke volume. Okay, and we've already discussed this. So venous return basically determines the amount of blood जो कि ventricle के पास है immediately prior to contraction. What do I mean by that is your end diastolic volume. We had a very great discussion on your cardiac cycle, and we discussed कि कैसे your end systolic volume and the end diastolic volume में से the stroke volume is derived, and what is remaining is your end systolic volume. We even discussed the phases of your filling of a ventricle. That is there were three phases, I think: rapid filling phase, slow filling phase, and the third one was your active filling phase, also known as the last filling phase. That's your mainly due to the atrial filling phase. You used to say that was coinciding with your atrial systole. So we have discussed that, and we know this end diastolic volume is going to be something known as a preload. And what is a preload? That before the contraction of the muscle, what is the amount of tension that is there in that muscle is your preload. Okay, and you have already seen how the initial length of the muscle fibers will contribute to something known as the Frank-Starling law, right? जिसमें हमने ये पढ़ा था that a muscle, when it achieves its length, which is known as its resting length, it's going to generate the maximum amount of active tension and the maximum amount of total tension. Okay, So just remember, end diastolic volume determines the preload on the heart. Okay? And the stretch on the heart wall due to the load to which a cardiac muscle is subjected before shortening is known as the preload. And after it does its contraction, what is it doing against? That will be known as the afterload. That is mainly determined by a total peripheral resistance. Okay, so let's just see what are the different factors which can regulate your venous return. We can have a skeletal muscle pump, very important. We can have a thoracic or a respiratory pump. The third one is the cardiac pump. Okay, that is the visceral turbo force. We have the blood volume and we have the ANS activity. We have some other factors as well, like your posture. It could be your venous resistance, your ventricular filling, as well as your ventricular compliance. So let's just discuss them. The first one that's very easy, that's your skeletal muscle pump. As we know, that basically this is the unit direction. What is happening is before muscle contraction, the blood is going to enter the vein, and when the muscle contract, it's going to squeeze the blood which is in the veins into your that is your receiving chamber. That's your right atrium. Okay, so that is your skeletal muscle pump. And how do we ensure the unidirectional flow? That is mainly due to your valves. We have these guarding valves जो कि prevent करेंगे your until or unless there is no venous incompetence. All right. So the second one is your thoracic pump. What are we discussing about thoracic pump? Whenever we do inspiration, the intrathoracic pressure is going to decrease, and that's going to generate a negative pressure, which is going to get transmitted to your great veins, okay, as well as to the atria. And what is that going to do? That negative pressure is going to pull the amount of venous return from the peripheral veins into your heart. So that is your second one. That is your thoracic pump. There is one more factor involved in this. Whenever we are going to inspire, that will also compress what? That's going to lead to compression of the abdominal veins as well. And that compression is also going to facilitate your venous return. So that is the second factor that is involved. That is your thoracic pump. Okay, I hope that's clear. The third one is your cardiac pump. इसमें हमारे पास दो forces हैं. The first is the viscer frontae force, and the second one is the viscer turgo force. What do I mean by a viscer frontae? So viscer frontae means it's a suction force, and a viscer turgo is a propelling force. That's the first difference. What is viscer frontae? Viscer frontae suggests that whenever there's going to be a ventricular systole, that's going to pull the AV valve downwards, as we've already seen, due to the Purkinje muscle की activity to prevent the overturning of the valves. It's going to pull the AV valves downwards, and इसके कारण atrial pressure is going to drop. On a JVP, that's known as the X dash or the X prime descent. Okay? So this negative pressure is going to be the one in the right atrium that's going to suck the walls of the veins together before they enter the chest. And इसके कारण सारा का सारा blood which is in the peripheral veins is going to go there. So in short, a viscer frontae force is a suction force. And what about a viscer turgo force? Viscer turgo force is very easy. कि हमारा अगर peripheral artery में whatever pressure that we have, that's going to get transmitted to the capillaries, that's going to be transmitted to the veins. And this pressure that was originally in the peripheral artery is going to be the one that's going to lead to venous return. Okay, so the propelling force which pushes the blood from the great veins into the right atrium, and how is it related to? It's mainly related to your arterial pressure. Okay, so that is your cardiac pump. What about the blood volume? We already know कि एक चीज होती है that is known as your mean circulatory filling pressure. Okay, what is that? कि whenever the flow of blood everywhere in the circulation, whenever it will cease, the amount of pressure that is in the arteries and the veins will come to an equilibrium, and that's known as the mean circulatory filling pressure. Okay, I hope that makes sense. We have two types of mean circulatory filling pressure. We can have a mean systemic filling pressure. Okay, so the pressure measured in the systemic circulation after the blood flow has been stopped, that is known as the mean systemic filling pressure. That is the PSF. And usually this PSF equals the mean circulatory filling pressure, and that amounts to be about 7 millimeters of mercury. Okay. So second thing is your blood volume. जितना ज़्यादा blood volume होगा, that's going to contribute to this mean circulatory filling pressure, which is normally about 7 millimeters of mercury. And this, what will this mean circulatory filling pressure do? That represents the pressure pushing venous blood from the periphery towards the heart. I hope that's clear. Okay, so PSF 7 millimeters of mercury, that is your blood volume जो contribute करता है. That's your mean circulatory filling pressure. What next? So we know whenever there's an increased blood volume, that's going to increase the PSF, and that's going to increase the venous return. That makes sense. So blood volume पे depend कर सकता है. ANS activity is very important because what will happen is these veins have alpha-1 receptors. So whenever sympathetic nerve system is going to get activated, it's going to activate this alpha-1 receptor, and we know alpha-1 receptor is GQ-linked receptor, and that's going to lead to vasoconstriction, or it would be appropriate to call it as venoconstriction. And whenever venoconstriction is going to be there, so the compliance of the veins is going to fall. And now what will the vein do? The vein will try to empty itself into the heart again. So that is known as that is due to your ANS activity. Okay. So we have discussed all of this. We have discussed about the cardiac suction effect. That's your vis frontae. And we have also discussed about the pressure imparted to the blood by cardiac contraction. That's your vis of frontae. Okay. That's a vis turgo. This is a vis of frontae. The sympathetic vasoconstrictor activity we've already seen by the alpha 1 receptors. The skeletal muscle pump we've already seen how it can influence it. The respiratory pump we've already seen how the negative pressure is going to facilitate your venous return. We have seen the blood volume and something known as the mean circulatory filling pressure, which normally is about 7 millimeters of mercury. Okay, I hope that's clear. And all of these are the factors which are going to influence your venous return. And your venous return is going to influence your end diastolic volume, and this end diastolic volume is going to influence your something known as stroke volume. जितनी ज़्यादा आपके end diastolic volume में, two things are going to be there. The stroke volume is going to increase by itself, obviously, more amount of blood for the ventricles to contract. And the second thing was your end diastolic volume is something known as the preload. And जितना ज़्यादा हमारा preload increase करेगा, according to Frank-Starling law, it's going to increase. There's going to be an increase in the tension of the muscle because the length of the muscle fiber is also altering. It's going to reach its resting muscle length, जहाँ पे उसका active amount of tension is going to be maximum. ठीक है? So what are the miscellaneous हमने already पढ़ लिया. So venous return, I can make a formula sort of sorts. कि whenever I'm going to talk about इस case में, so venous return is going to be your pressure differences. What are the pressure differences do we have? The mean systemic filling pressure minus your right atrium pressure divided by the resistance to the venous return. This amount of flow is known as a venous return. Okay, I hope that's clear. Right. So then we can have some factors which are going to affect your myocardial contractility. Okay, we've already seen. So we can have one thing that is known as heterometric regulation. We can have that's your according to the Frank-Starling law. What did I say? Whenever there's going to be an increase in the end diastolic volume, that's going to stretch the ventricular muscle fibers, और वो इतना stretch करेंगे that's going to reach their resting muscle length, जहाँ पे active tension is going to be maximum. We've already seen this. So this is the diagram that I'm saying. कि this is the Starling curve, or I also call it as a cardiac function curve. And here was an MCQ actually. कि your Starling curve or your cardiac function curve, जो basically what does it represent? It's mainly a length tension curve. Don't forget this. ठीक है? It's not a length pressure curve. It's a length tension curve. ठीक है? So just see कि जैसे जैसे हमारी end diastolic volume बढ़ रही है, उतनी हमारी stroke volume is also increasing because the contractility is further enhanced. ठीक है? Right. This was heterometric regulation. What about homeometric regulation? Homeometric का मतलब ये है that your length of the muscle fibers is not changing. So it's homeometric. It's not changing. Okay. So homeometric regulation, how can it be there? We can have something which can increase your contractility. That is a positive enotropic agents. Then we can have something which can decrease your cardiac contractility. That's known as negative enotropic agents. So let's see that. Sympathetic stimulation is a positive enotropic agent. I hope you all know that. We can have catecholamines, glucagon, thyroid hormone, even your caffeine, right? Caffeine is going to be a very important positive inotropic agent as well. Hypercalcemia, digitalis, we've already studied the mechanism behind digitalis, how it binds to the obein binding site at the sodium potassium ATPase, increases the intercellular calcium that is there to facilitate more amount of cardiac contraction. Okay? And increased heart rate. Didn't we study that? That is a Treppe phenomena, also known as the Bowditch effect. Jitna zyada hamara heart rate badega, two things are going to happen. First thing is cardiac contractility is going to increase, but just remember, whenever there's an increase in the heart rate, the amount of diastole time that we have, that's also going to decrease. And what will that do? That will decrease the end diastolic volume. And that might decrease the stroke volume. So even when there is an increase in heart rate, we are saying that there is increase in contractility. Don't forget its effect on the end diastolic volume as well. Okay? An increase in body temperature is also a positive inotropic agent as well. So let's see the negative ones. Parasympathetic stimulation, obviously. Then we have hypoxia, hypercapnia, acidosis. Direct cardiac depressant actions हैं इन तीनों के. Hyperkalemia, hypercalcemia, ठीक है. Quinidine, procainamide, barbiturates, ठीक है. ये हमारे sodium channel blockers हैं. That is quinidine, procainamide. And then we can have intrinsic depression in your heart failure as well, है ना? या फिर loss of myocardial function है, whether there's any cardiomyopathy or any myocardial diseases, myocarditis. So that is an intrinsic depression in your heart rate or your heart failure, mainly contractility is depressed. Okay? So we've already seen what are the effect of changes in the myocardial contractility on the Frank-Starling curve as well. Okay? Right. So these were the sympathetic nervous system effect. How can it do that? It's going to lead to increased calcium movement from the ECF to the cytosol. That's one mechanism it can do. Okay? How does it do that? Because हमें पता है that whenever we're going to have this beta 1 receptor, right? This is a G7 GS linked of receptor that's going to activate your adenine cyclase. That's going to generate your cyclic AMP. This cyclic AMP is going to activate your protein kinase A, and one of the proteins that's going to phosphorylate is your L type of calcium channel. Okay, and that's going to lead to increased amount of calcium influx, and that can lead to increase in contractility. That's one thing. Okay. The second thing we also studied was that it can also modulate your curve of your pre-potential that was in your slow action potentials. That is in your SA node and AV node. SA node and AV node mein, it can increase the slope of the pre-potential. Okay, increase in slope of pre-potential. And what will that do? That's going to lead to an increase in the heart rate. Okay, so that's going to lead to increase in heart rate. And how did we do that? बेसिकली हमने याद किया था दैट साइक्लिक एएमपी इज गोइंग टू ओपन दिस चैनल विच इज नोन एज योर HCN चैनल्स, और इसके कारण हमारा प्री पोटेंशियल ज़्यादा जल्दी कंप्लीट हो रहा था. दैट्स व्हाई वी आर गोइंग टू हैव अ पॉजिटिव क्रोनोट्रोपिक इफेक्ट. So we have seen that. Okay. So there are multiple mechanisms behind it. You can study all of them here. One is your increased calcium is moving from the ECF to the cytosol. That's one. There's increased calcium movement from the sarcoplasmic reticulum to the cytosol as well, because यहाँ पे भी calcium जो channel है यहाँ पे, that's your IP3 receptor or your ryanodine receptor, that's also modulated by your protein kinase. Then we can have increased rate of calcium dissociation from your troponin C, very important mechanism indeed. And we can have increased rate of calcium reuptake into your sarcoplasmic reticulum. How was this mediated? We studied about something known as phospholambin, and how phospholambin, when it is phosphorylated, it is inactive. Okay? And this phospholambin was the one which was inhibiting your circa. So now we have freed the circa from its inhibition. Now we can generate more amount of contractions in a given unit of time. Okay? Right. The last thing. So this is what you have seen. We've talked about the voltage-gated calcium channels as well. We've talked about the ryanodine receptors in the sarcoplasmic reticulum as well. We've discussed about phospholambin as well. And don't forget your troponin friend. Okay? So troponin, it's going to shorten the calcium troponin binding time and hence generate more amount of your cardiac contractions per unit time. Okay? All right. So we've seen all of this. So now let's discuss about the afterload now. So what is this afterload? Afterload refers to the force that the right and the left ventricles must overcome in order to eject blood to their respective arteries. And when do I mean by an afterload is mainly your total peripheral resistance. So what is the resistance present in the arterioles? Mainly because arterioles are your resistance vessels. So जो भी हमारे vasculature में जितनी resistance है, जिसके against we are doing this amount of work that is being done by the left ventricle and your right ventricle is known as your afterload. Okay, I hope that's clear. So it's largely determined by your blood pressure in the arteries of both the pulmonary and the systemic circuits. And how is it influenced by the total peripheral resistance? So the TPR is the determinant of your afterload. So whenever the afterload is going to increase, we're going to see a decrease in the stroke volume. Okay? Whenever the preload was increasing, we were seeing an increase in the stroke volume. Whenever the afterload is increasing, we are seeing a decrease in the stroke volume. Okay? Right. So that makes it clear. So let me just discuss some more principles behind it. All right. So let me just tell you something about your fixed principle. Okay. So this fixed principle is very important when we are going to evaluate and calculate your cardiac output. So what is this fixed principle? It's very easy. So it means by K, the simple thing is, right. So when I say that the amount of oxygen that is consumed by our body is about 250 ml per minute. Okay, that is the amount of oxygen that is being consumed by our body at and whole at the level of the lungs. So the oxygen that is entering in the lungs is 250 ml per minute. We have the venous side of the circulation. Okay, we have the venous side जहाँ पे हमारा जो amount of oxygen that is 140 ml per liter. Then we have the arterial side जहाँ पे हमारा oxygen has suddenly increased to 190 ml per liter. I hope that's clear. So this makes to be about 0.14 ml and this makes to be about 0.19 ml. Okay, whatever be the case, the oxygen is being added. And what is all facilitating that? The flow over present over here. So how can I calculate the flow which is here by having these three variables available to me? Okay, how do I do that is, I'm going to see that the amount of oxygen that is being removed by a particular organ, okay, divided by the arteriovenous difference between them. That's going to give me the flow. Let me repeat. So the amount of oxygen that is being extracted, that is being used, okay, by any organ, or that is being used by any organ. It could be oxygen or it could be anything else as well. So jitni amount of substrate jo bhi hai that the organ is either disposing of, okay, divided by the arteriovenous concentration of that particular amount of substrate, it's going to give me the blood flow that is going to be there. So when I calculate the blood flow through the lungs, this was in this example, I can say the Q is going to be the oxygen consumption, which I've already taken to be about 250 ml per minute, divided by the arteriovenous pressure, जो हमारे arteriovenous concentration जो है of oxygen में क्या difference है. That's going to give me the blood flow through the lungs. And this is the same mechanism that we even use for your PAH clearance as well. Then when उस time पे we were calculating something known as a renal plasma flow, और हमने same mechanism use किया था, ठीक है. Now we are using the same mechanism over here as well. Fixed principle is applicable in many different conditions. Okay. All right. So we've already seen this. So this is how the experiment was done. कि ऑक्सीजन कंजम्पशन हमें पता थी, हमने आर्टीरियल ब्लड में हमें ऑक्सीजन का कंसंट्रेशन पता था एंड मिक्स वीनस ब्लड में हमें ऑक्सीजन का कंसंट्रेशन पता था. इतना पता करने के कारण we can calculate everything. ठीक है. So that is the thing that is present. All right. There are other methods of measuring your cardiac output regarding except of the fixed principle. We're going to discuss that in detail, but abhi I thought ki, so let's see ek baar, again, fixed principle is defined as the amount of substance taken up or given by an organ per unit time. Okay? That is equal to the arteriovenous difference of the substance time the blood flow. They've already reversed it. Okay? ये कह रहे हैं कि ये amount of substance is equal to the flow times the difference. Or I can say, if I want to calculate the flow, then I can take the oxygen consumption divided by the arteriovenous difference in the oxygen concentration. Okay, so that it means the same thing. We can have other methods of your calculating your cardiac output. One includes your echocardiography, obviously. We have dye dilution method and we have the thermodilution method as well. Okay, so these are the different things that I can say. All right. Let me discuss something about the Laplace law. Okay, so there is one thing known as the Laplace law. What does this Laplace law say? We can use Laplace law in two different conditions. We can assume a sphere or we can use a cylinder. Okay. Next, when we're going to discuss the hemodynamics of blood flow, उस time पे we're going to assume a blood vessel to be a cylinder, and उस case में थोड़ा सा Laplace law will going to be changed. In case में we are assuming the heart to be a circle, or even in the case of alveoli, we take it to be a circle. So this case में the Laplace law states that your pressure is equal to your 2 times the tension times the thickness divided by a radius. All right, divided by a radius. So what are the ramifications of this? What do I mean is, whenever I'm going to increase the radius over here, okay, whenever I'm going to increase the radius over here, and whenever the tension is not changing, the pressure that I'm going to generate via this system is going to be reduced. You get it? Matlab simple hai. Whenever I'm going to have heart failure, jahan pe I have too much amount of preload, which the heart is not able to get rid of, it's going to increase the radius, it's going to increase the cardiac dimensions, and agar tension nahi change hoti hai, which obviously it will, we have something known as volume overload hypertrophy, but agar tension change nahi hoti hai, the pressure that is generated by that ventricle is going to be less. So that is what is known as the Laplace law in consideration of your CVS physiology. Okay? So whenever the preload is too much, the heart is under volume overload. उस केस में it undergoes volume overload hypertrophy to counteract the effects of your Laplace law. And how does it do that? What am I saying over here? What am I proposing? Whenever I have an increase in the radius, if I'm saying the tension is normal, then I'm going to see a decrease in the pressure. To avoid this decrease in pressure, what I'm going to do is, ये जितना radius increase किया, let me just try to increase the thickness of the cardiac muscle. Okay? So that I can increase your tension, and उसके कारण the pressure that I've generated remains normal. So वो एक mechanism है, that is your volume overload hypertrophy. Okay? So this is what I was saying. In cardiac failure, what is going to happen? Okay? We have already seen that. The heart becomes globular, there's an increase in the rate. With increase in rate, the pressure generated by the ventricles decreases. To manage the radius of the ventricle, filling of the ventricles can be reduced to decrease the volume work of the heart. And how do we do that? Us case mein, we give something known as venodilators, or we give something known as diuretics, which are going to decrease the blood volume, decrease the venous return, decrease the preload. Okay? So that is one thing, how we can counteract your cardiac failure and give all of these amount of different drugs, which can actually counteract the effects of your Laplace law, which were considered to be not good for us because it was decreasing, it was hampering the amount of pressure that was being generated by the ventricles. Okay? I hope that's clear. All right. ठीक है जी. Now let's move on to. So we've already seen the heterometric regulation. ठीक है? हमने heterometric regulation हमने बहुत पढ़ लिया था. That was a skeletal muscle pump आ जाएगा इसमें, हमारा cardiac pump आ जाएगा, thoracic pump आ जाएगा, blood volume, ANS activity, miscellaneous. We discussed all about the heterometric regulation as well. ठीक है? And heterometric regulation, obviously, whenever there's going to be an increase in the venous return, it's going to increase the end-diastolic volume. And what is that going to kick in? The Frank-Starling mechanism. And that is why I'm calling it as heterometric, because यहाँ पे length of the muscle fiber is going to increase. So whatever is increasing the venous return is bound to be one cause of your heterometric regulation of your cardiac output. Okay? And what about the homeometric? It was mainly sympathetic stimulation, or it could be parasympathetic stimulation. We discussed about the different factors, that is hypoxia, hypercapnia, acidosis, how it is negative inotropic, right? We discussed about hormones as well, like catecholamines, thyroid, and glucagon in particular. We discussed about the ions, that is hypercalcemia has a positive inotropic effect. We discussed about digitalis as well, how it has a positive inotropic effect. And we also discussed the force frequency relation, that is your Bowditch or a staircase phenomena. Whenever there's an increase in the heart rate, we're going to see positive inotropic effect. Okay? Right. So we have discussed all of this. Okay. So now, as we are done with your cardiac output, let me just summarize and say what are the measurement, how can we measure your cardiac output. The first one is your echocardiography. The second one is your direct fixed method, which you've already seen. And then we have the indicator dilution method, either thermo or dye dilution method. So let's just go over them one by one. So obviously, the first one is your echocardiography. So I hope that is not a discussion of ours. That's mainly covered will be in radiology. That's an echocardiography. But Abhi, we're going to go to the direct fixed method. And what was that? The amount of substance taken or given by an organ is equal to the blood flow times the difference in the arteriovenous concentration. Okay? And whenever I have to calculate this stuff, I'm going to divide it. The amount of the substance taken or given by an organ divided by the arteriovenous concentration. That's going to give me the cardiac output. Okay? So the amount of substance taken or given per minute divided by the arteriovenous concentration of that substance is going to give me the cardiac output. Okay. Same could be done for renal plasma flow, but then us case mein we are going to use something known as pH. And why only pH? Because if you remember, so sorry for the interruption. ठीक है, so we have already seen the direct fix method as well. तो हमें जो भी चाहिए था we can calculate उसके accordingly, and we have already seen this picture as well, and we have already used the example that we did in case of your lungs, that is 250 ml per minute wala, that is the oxygen consumption by the body, and we calculated your arterial concentration and we calculated your venous concentration, and हमने minus कर दिया था दोनों को, we obtained the difference, and now we have the cardiac output to be about 5 liters per minute, which we've already studied. ठीक है. But the disadvantages of your fix principle or your direct fix method is mainly it is an invasive process. You know? It's an invasive process and the patient may be anxious of catheterization. Okay? So we discussed some other methods as well. We discussed the dye dilution method. Okay? तो इसमें क्या था, एक known substance था, कोई भी amount of substance such as a dye or a radioactive isotope, you inject it into a large systemic vein, preferably into the right atrium. So what will happen, this indicator is going to pass rapidly through the right side of the heart into the lungs, then into the left side of the heart into the systemic arterial system. And now you basically do is successive serial samples may you just measure the amount of concentration that is being in the arterial blood. Okay, so the output of the heart that is going to be there, that's going to be the amount of indicator that you injected divided by the average concentration in the arterial blood after a single circulation throughout the body. ठीक है, so that is your dye dilution method, कि हमने जितना इंजेक्ट किया, मान लो 5 milligram we have injected, ठीक है, so there's going to be an increase in the heart is going to be increase in the concentration of the dye, and now it's going to be its equilibrium sort of, ठीक है, and then I can accordingly calculate कि कितना milligrams of dye I have injected, ठीक है, and divided by the average concentration of the dye and duration of the curve in your seconds, and that can be done. Okay, so that's your dye dilution method, right? And what should be the indicator की चीजें? Obviously it should be non-toxic, it should mix evenly, it should be relatively easy to measure its concentration, it should not alter the cardiac output, और basically कोई pharmacological substance नहीं होना चाहिए, है ना? And the most commonly dye that we use in this case is mainly your Evans blue, or we can use your radioactive isotopes as well. Okay. The second one that we have is thermodilution method, which is your cold saline wala. That is a cold normal saline we can take, and we can just measure कि कितना temperature change हुआ है in the blood. Okay. So that is your thermodilution method. All right. Okay. So now we are on to the main stuff, that is your regulation of your arterial blood pressure. Okay. So there are three mechanisms. We can have rapidly active mechanism, we can have intermediate active mechanism, we can have long-term active mechanism. And one of the long-term active mechanism, which is the only one in fact, that is your renin angiotensin aldosterone system, which you already discussed in great detail in your renal physiology. So today we are not going to discuss that. Mainly हमारा focus will be on the baroreceptor rapidly active mechanisms. Mainly we have the baroreceptor reflex, we have the chemoreceptor reflex, and we have the CNS ischemic response. Also we call it as the Cushing's ischemic response. The second one we are going to discuss is your intermediate active mechanisms. उसमें से you have the capillary fluid shift mechanism and your stress relaxation and reverse stress relaxation. So these are how do we modulate or how do we regulate the amount of blood pressure, whether it is increasing or decreasing. That's what we're going to study. So the first one that I'm going to discuss with you is the baroreceptors, which is the most important in fact. So let me just say, where are the baroreceptors located? So first one, we have three. We can have the carotid sinus में one. We can have at the aortic arch, and there is something known as cardiopulmonary baroreceptors. They are known as low pressure baroreceptors. Okay, they're known as low pressure baroreceptors, and they are present in the right atrium and the left atrium. Okay, so baroreceptors. But when we say baroreceptor, we mainly measure the high pressure baroreceptors, that is your carotid sinus and your aortic arch. तो आज उनकी बात करें basically. And what does it play a role in? Short-term control. Okay, rapidly active mechanisms के through. Let me just go over the circuitry behind it. So here, just at the junction of your ICA, internal carotid artery, and your ECA, external carotid artery, there's going to be a bump over here, which is known as your carotid sinus. Okay? And at the aortic arch, we're going to have the aortic arch, obviously. Okay? यहाँ पे हमारे baroreceptors located है, and these baroreceptors, if I'm going to show you the diagram actually, so basically कुछ नहीं है, the baroreceptors which are interspersed between your elastin fibers. Okay, so these are these organs which are interspersed within your elastin fibers present in the blood vessels themselves. Okay, it's not a different kind of a thing. It's going to intersperse into the blood vessels itself. So what's going to happen is, so whenever we're going to have an increase in the blood pressure, and MCQ के लिए the most potent stimulator of your baroreceptors is an increase in the pulse pressure. ठीक है, whenever more pulsatility से blood flow करेगा ना, whenever there is rapid fluctuations of blood pressure, वहाँ पे हमारी baroreceptor की activity maximum होती है, as opposed to something known as the sustained increase in your blood pressure. फॉर एग्जाम्पल क्रॉनिक हाइपरटेंसिव्स में ऐसा नहीं है कि उनका बायोरिसेप्टर हमेशा किक इन करके रहता है, दे बायोरिसेप्टर एडजस्ट टू इट, but the baroreceptor is more sensitive to fluctuations of blood pressure. So we are going to discuss mean as the pulse pressure and your mean arterial pressure. So whenever these guys are going to increase, whenever the blood pressure is going to increase, that's going to be sensed by the carotid sinus baroreceptor and aortic arch baroreceptors. Carotid sinus wala baroreceptor is supplied by your ninth nerve. That's known as the carotid sinus nerve. It's a branch. And the जो हमारा aortic arch वाला है, that is by your aortic depressor nerve. That's a branch of the vagus nerve. Okay? Both of them are going to transmit the signals into your one portion that's known as the nucleus of tractus solitarius. Okay? Nucleus of tractus solitarius. They're going to stimulate the NTS. These NTS neurons are glutaminergic neurons, but they're excitated neurons. These NTS neurons are going to do two things. They're going to, once of all, they're going to activate a region in your medulla known as the caudoventrolateral medulla, CVLM. Okay? Caudoventrolateral medulla. To be very frank, this caudoventrolateral medulla is nothing but it's like your parasympathetic nervous system ka center. Okay? This is your parasympathetic nervous system ka center technically. So this caudoventrolateral medulla is going to activate your parasympathetic nervous system ka center. That's mainly your nucleus ambiguous and dorsal nucleus of vagus. Okay? So this CVLM is activating nucleus ambiguous and dorsal nucleus of vagus, which are going to modulate their influences via the 10th nerve again. Okay? जो हम कहते हैं parasympathetic nerve supply to the heart. Okay? So one thing that I'm seeing is whenever there's an increase in the blood pressure, this parasympathetic system is getting activated. On the other hand, the CVLM, it's going to contain your GABAergic neurons. All right. So these are going to contain your GABAergic neurons, and they're going to inhibit your RVLM. That's your rostral ventrolateral medulla. They're going to inhibit your RVLM. That's your rostral ventrolateral medulla by releasing GABA. This RVLM neurons are now inhibited. And whenever these RVLM neurons are inhibited, it's going to inhibit your sympathetic neurons which are exiting from the intermediate lateral horn of the spinal cord. Okay. So what is happening? We're having a stimulation of the parasympathetic nervous system, and we're having a downregulation of your sympathetic nervous system whenever there's an increase in the blood pressure. And what are the effects that I will get? So whenever the baroreceptors are stimulated, I'm going to see a couple of things. First thing, the heart rate is going to decrease, the cardiac output is going to decrease. We might even have hypertension, okay, but that's not the right thing. Hypertension is not there. We can have venodilation and vasodilation because हमारे जो sympathetic nerve fibers हैं, they're also modulating your activity in your blood vessels as well. And mainly blood vessels are mainly supplied by alpha-1 receptors, except at your skeletal muscle there are beta-2 receptors. That's a beta-1 receptor, beta-2 receptor. That is different thing. But mainly it is alpha-1 receptor which is predominant. In this case, we're going to see vasodilation, venodilation, and we're going to see decrease in heart rate as well as a decrease in cardiac output. So this is the baroreceptor reflex. Okay. So just remember, these are more sensitive to pulsatile pressure as compared to constant pressure. And just remember, one thing is the threshold that is there. मतलब baroreceptor कब तक detect कर सकता है and कब तक fire कर सकता है कितने low pressure पे. Just remember, ये पूरा जो mechanism it can get reversed as well. Whenever I'm going to see a decrease in the blood pressure as well, the decreased activity of the baroreceptor is going to inhibit the inhibition of your sympathetic nervous system or inhibit your parasympathetic nervous system, where I can see an increase in the heart rate and increase in cardiac output as well whenever the blood pressure is less. Till what pressure does it operate, this baroreceptor mechanism? At 50 से लेके 60 millimeters of mercury. Okay? 50 से लेके 60. If I have to be very honest, just remember 60 millimeters of mercury. Okay? That's the one thing. All right. So this is the baroreceptor mechanism, and we have seen the structures as well. Okay. We discussed something about the cardiopulmonary receptors as well. जो हमने कहा था atria में mainly present है. We can have atrial stretch receptors. We can have ventricular stretch receptors as well. Okay. And one of the atrial stretch receptors हमने already पढ़ लिया है via the Bainbridge reflex. Okay. So there are two types of atrial stretch receptors. We can have A type and we can have B type. The A type stretch receptor are going to discharge during atrial systole, and the B type atrial receptors are going to discharge at the peak of atrial filling. Okay, so थोड़ा सा difference है. तो यहाँ पे venous return यहाँ पे ज़्यादा stimulate करेगा B type of atrial stretch receptors को. Whatever be the case, whenever there is increased amount of stretch in the atria, whether it be during atrial systole or during atrial diastole due to venous return, what's going to happen? First thing, via the Bainbridge reflex, by locally modulating a SA node की activity, by increasing the SA node की activity, we're going to obviously seeing an increase in the heart rate. But just remember, it's also going to mediate vasodilation, and we're going to see a decrease in the blood pressure. Okay, so cardiopulmonary receptors are a tad bit different from your normal baroreceptors. Baroreceptors were decreasing your blood pressure as well as decreasing your heart rate. These stretch receptors via the Bainbridge reflex might increase your heart rate. Okay, so that's a very important thing. But the ventricular जो हमारे stretch receptors है ना, they're going to do the exact deposition that is present in your baroreceptor. They're going to decrease the heart rate, decrease the blood pressure. Okay, so हमारे cardiopulmonary receptors है, they are your low pressure receptors as compared to your baroreceptors. Okay, so the second mechanism that I have for you is something known as the Bisol's Irish reflex. Okay, so what is this Bisol's Irish reflex? It's basically, it's not a physiological response. एक दो pathological conditions हैं which can exist जहाँ पे it can happen. Okay, so what is this Bisol's Irish? We're going to see some kind of nociceptive, I would say. कोई ना कोई ऐसा stimulus है which is very disagreeable to our body. So let's see what are those stimuluses which can stimulate this reflex. One thing is capsaicin. Okay, very hot spicy foods. We can have 5-hydroxytryptamine. Very important, I would say, that's the most important. That is an MI and reperfusion injury. Okay, whenever the person is going to suffer from myocardial infarction, we're going to see this Bisol's Irish reflex as a part of your reperfusion injury and toxic chemicals. So all of these are going to stimulate your Bisol's Irish reflex. Okay, and how are they mediated? We have some fibers known as your chemosensitive vagal C fibers, which are present in the cardiopulmonary regions. Okay, to be very specific, what are the regions जहाँ पे हमारे Bisol's Irish के हमारे chemoreceptors present हैं? We have the alveoli, the ventricles, the atria, the pulmonary artery, as well as the great veins. Okay, ठीक है. So whenever this is stimulated, whenever these chemosensitive C type of vagal fibers जो हैं, they're going to activate your CVLM, that is cordoventrolateral medulla. And what was the CVLM doing? It was inhibiting your RVLM, and ultimately जो इसका मैकेनिज्म है, इसके जो चीजें होंगी, what we're going to have a decrease in the heart rate, we're going to have a decrease in the blood pressure, as well as we're going to have apnea in the form of rapid shallow breathing. So this is known as the Bisol's Irish reflex. Okay, it might even lead to a vasovagal syncope. So one of the reasons or one of the triggers for your vasovagal syncope might be emotions, it might be increased amount of spicy food, any kind of nociceptive stimulus. So all of that is mediated, all of that hypertension that occurs in your vasovagal syncope is mediated by the Bisol's Irish reflex. Okay, very important. So the next reflex I have for you is your chemoreceptors. Now usually chemoreceptors, we usually study it in your regulation of respiration. यहाँ पे I'm going to study only the effect of the chemoreceptors on your mainly your cardiovascular system. We're not going to discuss the effect of your chemoreceptors और जो भी हम central chemoreceptors के बारे में पढ़ते हैं, we're not going to discuss all of that. ठीक है? So whenever the chemoreceptors are stimulated, I just want you to remember it's going to increase the vagal activity. बात खत्म. तो heart rate भी कम होगा and contractility भी कम होगी. But what are the different things which can actually stimulate your chemoreceptors? So let's just try to see that. So there are three main potent stimulus of your chemoreceptors in decreasing order. So the first one is your low PO2. I'm going to say hypoxia. Okay? So whenever the partial pressure of oxygen is going to drop below 60 millimeters of mercury, that's the strongest stimulus of your chemosensitive chemoreceptors. The second one is your PCO2, hypercapnia, being more than 40 millimeters of mercury, and your acidosis, that is pH being less than 7.35. And how is it all mediated? Especially the oxygen wala portion is, this low amount of oxygen is going to close these, actually, chemoreceptors may have something known as a glomus cell, okay? And we are supporting cells known as the sustatenticular cells. तो यहाँ पे glomus cell की हम बात कर रहे हैं. Whenever the oxygen low, oxygen tension कम होगी locally in the glomus cell अगर कम होगी, it's going to close this potassium channel, which is oxygen sensitive. I've already told you guys, whenever a channel is sensitive, then that means it's going to be inhibited by it. If it's an oxygen sensitive potassium channel, it's going to be inhibited by oxygen. Whenever it's an ATP sensitive potassium channel, it's going to be inhibited by ATP in case of insulin release. तो यहाँ पे oxygen अगर कम है, then what's going to happen is we're going to have closure of the oxygen sensitive potassium channel. We're going to have depolarization of the glomus cell. And what's going to happen is जैसे depolarization होएगा, voltage gated calcium channels are going to open. We're going to see increased amount of calcium coming in. और जो भी हमारे यहाँ पे nerves हैं, which are going to carry from the chemoreceptors, which is mainly your 10th nerve, that's going to get stimulated. So the main thing that is carrying is हमारे chemoreceptors यहाँ पे present है, peripheral chemoreceptor. They're either present at the carotid sinus or they're present again at the aortic arch. Okay? Carotid sinus is again supplied by the 9th nerve. The aortic arch is again supplied by the 10th nerve. And this branch of the 9th nerve is known as the Herring's nerve. Okay? So this is the Herring's nerve. So this is the chemoreceptors wala portion. What do? They decrease the heart rate, they decrease the contractility. We are not discussing the regulation. That is a chemical regulation via respiration. That will be covered in respiratory physiology. Okay? We have discussed this. Then the last one that is actually remaining is your CNS ischemic response. Okay? So just remember, हमारा जो ये सारे जो rapidly active mechanisms हैं, they are mainly in three brackets, I would say. Whenever the pressure up to 60 millimeters of mercury, the baroreceptors can function. Done. I told you that the threshold for your baroreceptor is 60. Whenever the pressure is going to decrease less than 60, then some other things are going to function. And the last response that we have is known as the CNS ischemic response, which is going to occur whenever the pressure drops to even 30 millimeters of mercury. So that's a very less amount of CNS ischemic response. So that's why it's known as the last ditch stand of your body. And how is it mediated? So let's just see that. So up to 60, we have studied the baroreceptors can control it. 30 to 60, your chemoreceptors can control it. Done. But whenever it is less than 30 millimeters of mercury, we're going to have the CNS ischemic response kicking in. Okay, the CNS ischemic response, I would say, actually we have two reflexes which go by the same name. थोड़ा सा difference है उनमें. One is known as the Cushing's reflex and one is known as the CNS ischemic response. दोनों का mechanism almost same है, but the reasons might be different. Cushing reflex का main reason is increase in ICP. The CNS ischemic response का mainly the main reason is central hyperperfusion. मतलब किसके कारण blood flow अगर कम हो रहा है directly. It could be anything. It could be an atheroma, could be anything, could be hemorrhage. ठीक है, whatever. तो mechanism दोनों का same क्या है? Whenever we're going to have CNS ischemia, जो हमारा rostroventrolateral medulla है, that is very prone to hypoxia. And whenever it undergoes hypoxia, it's going to stimulate it. मतलब वो vasomotor center जो है RVL में, it's going to get stimulated, and that's going to increase the blood pressure. And we've already seen whenever the blood pressure is going to increase, that's going to what? That's going to kick in your baroreceptors, and उसके कारण we are going to see a decrease in the heart rate. Right? We're going to see a decrease in the heart rate. And the last thing that might be affected in this case, because the RVLM is very close, that's present in the medulla, it's very close to some centers which modulate respiration. We're going to see irregular respiration in this as a part of it. We're going to see three responses in the Cushing reflex. The first one is bradycardia, mediated by the baroreceptors. We have hypertension, that is due to the RVLM, which was suffering the hypoxia because it's very sensitive to hypoxia. And the last one is your irregular respiration, that is bradypnea. Okay. If it is mediated due to increase in ICP, then that's known as Cushing reflex. If it is due to decrease in the blood flow to your that's through your cerebral vessels, then that is known as a CNS ischemic response. Just remember, it's a last bit stand, triggers at less than 30 millimeters of mercury. Okay, all. Then we have the intermediate regulatory mechanisms. उसमें हमारे पास दो हैं. One is capillary fluid shift, which is very easy. वो क्या कह रहे हैं? Whenever we are going to have a decrease in the blood pressure, then that's going to decrease your PC. What was PC? That is the hydrostatic pressure present in the capillary. Usually this hydrostatic pressure present in the capillary was facilitating filtration. But now because the PC has decreased, what are we going to have? We're going to have the opposite. The osmotic pressure is going to become more than the hydrostatic pressure. And now instead of filtration, we're going to have reabsorption of fluid. That's the main thing. That's known as what? That's known as a capillary fluid shift. It's an intermediate regulation of your blood pressure. Okay? So whenever the BP decreases, the decrease in hydrostatic pressure is a movement of fluid from the interstitium into the capillaries. And that's going to increase the blood volume. That might restore your blood pressure. Okay? That's what we're going for. The second one is a stress relaxation. What do I mean by that? The stress relaxation. Stress relaxation का मतलब ये है, whenever I'm going to distend a vessel, ठीक है? Whenever a vessel is going to distend, initially उस distension के कारण और pressure के कारण उस vessel का भी pressure जो है that's going to increase, ठीक है? Till a point. उसके बाद क्या होगा? Now we're going to have stretching of the smooth muscles which are present in the vessel wall and वो dilate करेगी, ठीक है? And उसके कारण उसकी compliance जो है that will going to increase and now the pressure is going to fall. So what are we seeing? So initially I'm seeing a spike in the blood pressure, and now I'm seeing all right. So we've already discussed the rapidly acting mechanism of your blood pressure control, as well as already studied the intermediate acting mechanism of your blood pressure control. The long-term wala, that is your renin-angiotensin-aldosterone system, we've already covered in renal physiology, so we will not be going into the details of it. Okay, so we'll be skipping that portion. You can refer to your renal physiology for that. And the last thing we've already seen is your autoregulation. One was that I studied in renal physiology was again your myogenic reflex, or they can have key to blood flow to any amount of organ that can be regulated. Okay, via metabolic needs. For example, whenever I'm going to have a decrease in the oxygen, hypoxia that is, or I'm going to see a decrease in the pH, that is acidosis, or an increase in temperature, or an increase in potassium, or an increase in lactate, or adenosine or ADP, all of them are going to lead to vasodilation, and that will control the local amount of blood flow that is going there. Okay? And what is vasoconstriction mediated by? Mainly it is mediated by 5-hydroxytryptamine or your decrease in your temperature. And this 5-hydroxytryptamine might be released when your platelets are getting activated, right? Okay? So that via the dense granules, we've already seen that. Okay? And just remember, we have something endothelium, we have something known as the nitric oxide. Okay? So the endothelium is going to have something known as nitric oxide synthase. We have three subtypes of nitric oxide synthase. एक हमारा CNS में present है, दूसरा हमारा मैक्रोफेज में प्रेजेंट है, which is activated by cytokines that will generate your nitric oxide species, oxygen radicals, and that's going to facilitate your killing of your microbes. The third wala, which I'm going to be most important, is your endothelial cells में जो enos present है, that is known as endothelial nitric oxide synthase. ठीक है? And what is going to increase that nitric oxide synthase की activity is mainly an increase in calcium, very importantly, acetylcholine or bradykinin. Okay? They're going to stimulate your NOS. And what is this NOS going to do? Going to convert your arginine into citrulline while generating something known as nitric oxide. Okay? So this nitric oxide is also known as endothelial derived releasing relaxing factor, EDRF. Okay? And what is this NO going to do? NO is going to diffuse from the endothelial cell through the smooth muscle cell, and it's going to activate guanyl cyclase. Okay? So this guanyl cyclase, as we already studied in smooth muscle physiology, that's in nerve muscle, this guanyl cyclase is going to increase the amount of cyclic GMP present. That's going to activate your protein kinase G. And in this case, what are we doing is we are activating an enzyme known as myosin light chain phosphatase. I told you phosphatases are active when they're phosphorylated. So this protein kinase G is going to phosphorylate this MLCP, and that's going to make it active. And now it's going to cleave the myosin की जो भी light chains present है. And what will that lead to? That will lead to smooth muscle relaxation. So अभी हमने study किया है कि NO जो है that's mainly leading to vasodilation, smooth muscle relaxation. And how we already seen that, हमने देख लिया कि कैसे it mediates that. ठीक है? Then we have something known as endothelins as well. ठीक है? So we have something known as endothelins. So what are these endothelins is? All right. So endothelins, they're very potent vasoconstrictors in our body, and they work via the endothelin type A receptors, which is a GQ-linked receptor. Okay. And what are they increased or decreased by? Mainly the endothelins are increased by angiotensin 2 or catecholamines, hypoxia, insulin, HDL, oxidized LDL, वगैरह. And they're mainly decreased by everything that is mediating vasodilation, whether it be nitric oxide, your atrial natriuretic peptide, or your PGI2, that is prostacyclin. Okay? Just remember that. All right? That's your endothelins. Then we have something known as urotensin. It's a very important MCQ. It's the most potent vasoconstrictor which is present in our body. What is that? Urotensin. Very potent one is endothelin as well, but urotensin is the most important one. Okay? And it has been found that it's increased in hypertension as well as heart failure as well. Urotensin. Okay? And the last we can have something known as kinins. We've already discussed something about your bradykinin when we discussed about blood physiology. So we're not going to discuss that. Okay? So this is about your metabolic control. That's local control. So let's just see over here. This diagram I've taken from Guyton. So we can see कि कहाँ पे हमारे कौन-कौन से reflexes काम कर रहे हैं. And we can also see कि how much amount of time, milliseconds, minutes, hours, and days they're going to be triggered by. If you see baroreceptors, the CNS ischemic response and your chemoreceptors, they're mainly functioning in seconds. तो हमारे immediate mechanisms में आ जाएंगे. The stress relaxation and your capillary fluid shift वगैरह जो है, they're mainly intermediate. And then last में your aldosterone and your renal blood pressure control जो है mainly aldosterone के and RAS के through, that mainly is in days, and that's known as your long-term control of your blood pressure control. ठीक है? Right. And as we discussed about the feedback control as well, so this baroreceptor is a type of a negative feedback. But when we were discussing about this in endocrinology, we discussed something known as a gain. Okay, so just reminding you कि gain, and I told you guys कि उस time पे ज़्यादातर जो examiners देते हैं question about this particular topic about negative feedback is by taking the example of baroreceptors. So don't forget that. So gain is what? It's the correction that is done by the feedback mechanism divided by the residual error. So you can solve this MCQ. All right, and just check yourself whether you understand about the gain and the negative feedback. Okay, all right. Okay, ji. We have already studied the mechanisms. Now, thoda sa brief overview about the histology of the cardiovascular system. Okay, so let's just see something about it. As we already know, what are the different layers of a proper artery, I would say. So let's just see an artery pehle, and then we're going to discuss each and every bit of your vasculature which is left. Okay, so let's see what are the different portions. We have the outermost adventitia. उसकी नीचे भी दोनों के बीच में पहले हमारा है एक्सटर्नल इलास्टिक लमाइना, देन यू हैव द इंटरनल इलास्टिक लमाइना, एंड उसके बीच में वी हैव द पैक्ड लेयर ऑफ योर मस्कुलर लेयर. ठीक है, मेक सेंस. एंड इसके बाद फॉलोइंग दैट वी हैव समथिंग नोन एज योर सब एंडोथेलियल कनेक्टिव टिश्यू, देन वी हैव द बेसमेंट मेम्ब्रेन, basal lamina, and then you have the endothelium. So these are the different portions of your blood vessel. It could be mainly in case of an artery that we are saying. So what are the intima composed of? You know, it's composed of the endothelium. Endothelium. It's composed of a basement membrane, that's your basal lamina, and it is composed of your subendothelial connective tissue. Okay. Then we studied about the internal elastic lamina and the external elastic lamina. और दोनों के बीच में क्या हमारा muscular layer है. And muscular layer also contains some amount of elastin fibers, some amount of collagen fibers. ठीक है. And after the external elastic lamina, Then what do we have? We have something known as the connective tissue layer, which is your adventitia by all aspects. So what does the adventitia have? It has many amount of connective tissue. That's your collagen, elastic fibers, fibroblasts as well. It has its own blood supply. So just remember, जो हमारा outer one third of your artery is mainly supplied by your vasa vasorum, and the inner two third जो है हमारा, it's mainly supplied by what? It's mainly supplied by the blood itself, which is in that artery. Okay? So the vasa vasorum is going to supply the outer part of the artery. Then we have the lymphatics in the adventitia, and we have the sympathetic supply in the adventitia. Just don't forget that parasympathetic supply to the artery is not there. ठीक है, पैरासिम्पैथेटिक वैस्कुलेचर को ज्यादा मॉड्यूलेट नहीं करता है. So this is a typical example of your artery का histology. So let's just see एक एक का difference. So we have something known as large arteries. ठीक है, what are they? Aorta, pulmonary artery, and its major branches are known as large arteries. ठीक है, इनकी स्पेशल बात क्या है? They're very elastic. Elastic का मतलब whenever systole is going to occur in the ventricle, what do I see? I'm going to see distension of the aorta. ठीक है, now whenever the systole is going to end now, what I'm going to have? I'm going to have ventricular diastole. Okay, the ventricle pressure is going to reduce now. So whenever the pressure is going to reduce, won't the blood that is being propelled forwards have a tendency to return back? To prevent that from happening, what do we do is, this artery which is initially distended is now going to collapse upon itself and generate a recoil pressure. During which phase? During the diastole. So when I say the diastolic blood pressure is mainly mediated by the recoil effect of these large arteries. And what do I call this effect? I call this effect as the Wien-Kiesel effect. Okay, so this Wien-Kiesel effect, और इसके कारण large arteries are also known as Wien-Kessel arteries. ठीक है, so large arteries का mainly जो architecture it remains same, and mainly यहाँ पे मैं कोई आपको याद ये रखना है that there is going to be alternate arrangement of elastic lamina and smooth muscle layer. So it's like internal elastic lamina, smooth muscle, elastic lamina, smooth muscle, elastic lamina, smooth muscle, up to how many times? Up to 40 to 70 times. तो इतने ज़्यादा elastic ये vessels हैं. ठीक है, all right. The second that I'm going to discuss is about your muscular arteries. Okay, they are also known as a distributive artery. They're going to distribute the amount of blood that is there to the different peripheries. इनमें और उनमें क्या फर्क है? Mainly it has one internal elastic lamina, it has one external elastic lamina, it has one muscular layer, and that's it. It's not going to have alternate layers of your internal elastic lamina followed by your muscle layer. Okay, 40 to 70 times नहीं है, it's going to be only one time. So this is the example that I used earlier. So the muscular artery हमारा main जो artery का जो हम diagram बनाते हैं, that's your muscular artery. Internal elastic lamina, external elastic lamina, और दोनों के बीच में sandwich is your smooth muscle cell layer. ठीक है? And now the next one that we have is your arterioles. So arterioles are also known as your resistance arteries. Why so? If you see its lumen and if you see its wall thickness, ठीक है, कभी भी you not see that इतनी ज़्यादा जो lumen is to wall is to lumen ratio है, that's going to be maximum in case of your arterioles. And you can see the smooth muscle cell are very dense है. ठीक है, so it has the densest amount of smooth muscle cells. It has the maximum amount of wall lumen ratio as well. ठीक है, मतलब इसका ये है, and it's not even going to have any elastic tissues. ठीक है, so whenever it's going to vasoconstrict, it's going to be the main reason which is responsible for your afterload, and that is the reason why I call them as the resistance arteries. ठीक है, just remember according to Pausili's law, जो आपका resistance होता है, that is inversely proportional to your radius 4 times. ठीक है, very important. तो whenever it's an MCQ like sort of कि अगर resistance जो है मेरा अगर मेरा radius जो है decreases by 2 times, then a resistance is going to increase by 16 times, that 2 is to 4. ठीक है, so this is the thing. Arterioles जो है they measure about 10 से लेके 100 microns, and they're going to determine the blood flow to the tissue by serving at the resistance arteries, by contributing to something known as your afterload, by contributing to something known as your diastolic blood pressure. Okay, all of that can be happening. What about these meta-arterioles? Okay, for that we need to understand what is this meta-arteriole, pre-capillary sphincters, and all of this that is happening. So I would like to use this diagram. So what I am having is एक arteriole है मेरे पास, and I have something known as the meta-arteriole, okay, and I have something known as the capillary bed, okay. So this pre-capillary sphincter, okay, whenever it's going to contract, it's going to seize the blood supply which is in the capillary bed and facilitate all of that blood to go through the shunt vessel which is known as the meta-arteriole. Okay, it's going to be the meta-arteriole. Do you understand? So whenever this pre-capillary sphincters, usually they are partially contracted, and hence इतना भी ज़्यादा capillary blood नहीं present होता है, मतलब इतना भी ज़्यादा perfuse नहीं होता है any organ. Okay, whenever this pre-capillary sphincters are going to constrict now, all of that blood is going to be shunted away from the tissue and directly into this thoroughfare channel into the veins, okay, without any tissue exchange, right? And whenever this pre-capillary sphincters are relaxed, it's going to mediate all of that blood to go into the capillary bed and perfuse the tissue. ठीक है. So meta arterioles, I hope you remember, I hope you now understand what is the role of this meta arterioles, thoroughfare channel, and your precapillary sphincter. Whenever the precapillary sphincters are constricted, it's going to lead to all of that blood flowing through the meta arterioles and your thoroughfare channel and not having to undergo any exchange with the tissue. Okay. And whenever this precapillary sphincters are dilated, it's going to facilitate that amount of blood to go into the tissue and participate in exchange. Okay. So what are the things which can actually moderate the precapillary sphincters? Are the exact similar things जो हमने blood flow में पढ़ा था, that we were studying about the metabolic control of blood flow. What were they? Increase in temperature, hypercapnia, lactate increase होना, acidosis, increase in adenosine, increase in ADP. Okay, all of them are going to lead to relaxation of the precapillary sphincters, and that is the mechanism how do they increase the blood flow through the capillary bed. Okay, so you discussed about the metaarterioles. They're going to serve as vascular shunts when the precapillary sphincters are closed, mind you. Okay, all right. What about the capillaries? The capillaries, by all instances, are exchange vessels. So, इनके lumen का size is about 8 to 10 microns. Okay, we have three types of capillaries. We can have continuous capillaries, we can have fenestrated capillaries, and we can have sinusoidal capillaries. Okay, so the simplest of them all, I would say, is the sinusoidal capillaries. This हमारे endothelial cells हैं, and इतनी ज़्यादा distance है. They have a very big amount of interendothelial distances, about very high. That's about very big interendothelial gaps हैं इनके case में. And even if you see the basal lamina, that's your basement membrane, वो discontinuous है. That's known as the sinusoidal capillaries. What are the example? Mainly it's the spleen, or I can say the liver sinusoids, or I can say the bone marrow में जो sinusoids थे, तभी तो megakaryocyte enter करेगा इसमें, ठीक है? Or I can say the lymph nodes, वहाँ पे भी हमारे sinusoids थे, ठीक है? All of them are your sinusoidal capillaries. The second one I would say are your fenestrated capillaries. How are they different? They have these holes present within them, ठीक है? They are known as fenestrations, and they're going to participate in something known as filtration, कि जिसके through blood is going to filter through these pores. Okay, and what is the example of a fenestrated capillaries? I would like you to remember that's mainly the glomerular capillaries. Okay, so the glomerular capillaries are example of a fenestrated capillaries. And what about your continuous capillaries? Continuous capillaries में mainly इनके बीच में you have tight junctions. Okay, so you have a continuous basal lamina and you have tight junctions between them. Okay, but it might also contain some amount of pinocytotic vesicle जिसके through we can participate in endocytosis and exocytosis, and उसके through हमारा exchange होता है through these continuous capillaries. ठीक है, mainly they're present in the CNS, the muscles, lungs, and your exocrine glands. Okay, I hope I'm clear. All right. And the last thing, as you've already seen, that's your veins. Veins are also known as a capacitance vessel. मतलब they're जो maximum almost 50 से लेके 70% of your blood, circulating blood volume, is mainly contained in the veins. That is why we call them as the capacitance vessels. ठीक है? They have very sluggish amount of blood flow and they hold a बहुत ज़्यादा amount of blood. ठीक है? If you talk about the layers, the tunica adventitia in this case is the thickest layer. Usually it is the tunica media which is the thickest layer. In this case, the tunica adventitia is the thickest layer. They don't have an internal elastic lamina. They have a thin walled but larger lumen, and there are going to be valves which are going to be present, and they don't have a well-defined tunica media. Okay, very important. We have discussed about the histology. So these are the type of blood vessels that I've discussed. We have discussed the Windkessel effect, that is the aorta, the pulmonary artery, and its major arteries. And what are they characterized by? They're characterized by the Windkessel's effect. Then we discussed about the resistance vessel, that was the arterioles, okay, and we talked about the Poiseuille's law as well. We discussed about the exchange vessel, that is your capillaries, okay, and they were controlled by the precapillary sphincters and the metaarteriole and the thoroughfare channel. वो भी हमने discuss किया. We have the capacitance vessel, that were your veins, okay, very important. All right. So now let me just discuss something about hemodynamics of blood flow, okay, and that will be our last topic, okay. So just remember, पहला law that I would like to discuss is something known as the Darcy's law. The Darcy's law is very simple, बहुत simple है. That mainly states, I would say, we can remember it as V is equal to IR, right? We have studied the Ohm's law. If we say the I to be the flow and the resistance to be the resistance and the V to be the pressure changes, we can actually equilibrate and derive this Darcy's law. What does that state? The flow through a blood vessel is equal to the pressure gradient जो कि present है between A and B points divided by a resistance. That's very equivalent to your Ohm's law. So this law is known as the Darcy's law. ठीक है? If I talk about resistance, then I can use this one. अगर मैं resistance का उल्टा word use करना चाहता हूँ, then I can even say your conductance. So we have something known as hydraulic conductance. We have something known as your hydraulic resistance. So if I talk about cardiac output, which is a form of a flow, I can say the cardiac output is equal to your pressure in the aorta minus your central venous pressure divided by a total peripheral resistance. Very important. And because the central venous pressure is almost equal to zero, मतलब not exactly zero, but close to your P ATM, जिसके through हम compare कर रहे हैं, so the pressure in the artery can be equilibrated to be cardiac output time total peripheral resistance, and that is what we discuss. Okay, so the pressure that is present, जो blood pressure होता है, that is your cardiac output and total peripheral resistance का multiplication factor. ठीक है. The second theory जो मैं discuss करना चाहता हूँ, that is about the Bernoulli's theory, and we already studied in physiology as well. तो यहाँ पे क्या था? Only there was one thing that was playing a role, that was pressure. Bernoulli's theory में there are actually three types of energies which are there. There is something known as the pressure energy. ठीक है. There is something known as the potential energy, and there is something known as the kinetic energy. And all of these energies are interchangeable. ठीक है. What do I say? ये तीनों are going to make up something known as mechanical energy. ठीक है. These are components of your mechanical energy, and I can calculate through this equation कि अगर मेरा point A है और मेरा point B है, I can say the energy loss between them is going to be the difference in their mechanical energy. Why I'm so focusing on that is very simple, and you're going to love this principle actually. Okay, so we've seen the pressure energy, we've seen the kinetic energy, we're going to see the potential energy change, and they're going to be कि energy per unit volume जितनी before थी is going to be the energy per unit volume after through a flow, whatever that is happening. What do I mean by that is, मान लो यहाँ पे हमारा, so we have the pressure energy, kinetic energy, and the PGH, that is your potential energy. So what's going to happen is here, right? तो यहाँ पे what's going to happen is whenever I see यहाँ पे अगर मेरा lumen का diameter अगर कम हो जाता है, ठीक है, whenever I'm going to have a decrease in the lumen diameter, what I'm going to see, all right. So what's going to happen is this amount of kinetic energy जो पहले हमारी present थी और हमारी pressure energy and potential energy हमारी present थी, they're going to interchange now, ठीक है. So what am I going to have? Whenever this is going to decrease, the pressure might decrease. मतलब जो pressure energy जो है यहाँ पे it might decrease, but that is compensated by an increase in the kinetic energy. As I said, all of energy is interchangeable. तो हमारी जो pressure energy है वो कम हो गई है चाहे, but हमारी kinetic energy will take its place. So that is the reason why कि whenever I go from a larger lumen to a smaller lumen, two things are going to happen. The pressure might decrease, but what will happen to your kinetic energy? The velocity is going to increase. ठीक है. And that is the reason why कि whenever एक small orifice होता है, the amount of blood, the amount of anything, that is any fluid which is flowing through that small orifice, which was earlier flowing through a larger lumen, is going to go with a much higher amount of velocity. ठीक है. But the pressure at the smaller orifice will be less as compared to the larger lumen. ठीक है. So we already seen कि अगर P1 और P2 में अगर हम compare करें, the P2 जो है is going to be definitely less than the P1. But just remember the flow velocity through P2 is going to be much higher than your flow velocity to P1. That's your V1. ठीक है. Just remember that. ठीक है. So applications क्या है कि blood flow through a stenosed artery, what's going to happen is we're going to have an increase in velocity जो होगा as kinetic energy is going to be the decrease in pressure energy that is being generated. So this is your Bernoulli's theorem. ठीक है. ठीक है. Hemodynamics में this is very important. And because we discussed about the something with the potential energy, इसके कारण हमें एक और चीज बड़ी important मिल जाएगी, that if I say the heart and I calculate your mean arterial pressure to be 95 millimeters of mercury. Okay, I'm going to tell you how do we calculate the mean arterial pressure in case of an aorta. Okay, so let's assume the mean arterial pressure is 95 millimeters of mercury, and we discuss कि mechanical energy का एक form है known as the potential energy, and that is depicted as rho gh. ठीक है, where g is the gravitational constant and h is the height. So whenever I go above the heart level, what's going to happen is हमारा potential energy is going to fall, and whenever I go below the heart level, my potential energy is going to increase. My potential energy is going to increase. So that is the reason why when I say the MAP at the proximal aorta is 95 millimeters of mercury, but the MAP at the dorsalis pedis is about 183 millimeters of mercury, because as I go down, there's going to be an increase in the potential energy. Okay? And when I go up, there's going to be a decrease in the potential energy. Okay? Alright. Okay, now we're going to discuss about two flows, that we have the laminar flow and we have something with the turbulent flow. What do I mean by a laminar flow is, I'm going to have a blood vessel here and I see a fluid here. So according to the laminar flow, it says कि हमारा एक layer of fluid यहाँ पे होगा, and the subsequent layer of fluid which is on top of it is going to be there, and then we're going to have another layer, and we're going to have multiple layers stacked like this. Okay? So what do I mean by that is, with this जो ये हमारी first layer है, that's going to be have the least amount of velocity. Why? Because it is interacting with your lumen, जो हमारी vessel wall है. ठीक है? That's going to interact with your vessel wall. उसके कारण shear stress जो generate होएगा, उसके कारण उसकी velocity कम है. But the subsequent layer is going to glide over this, and उसके ऊपर वाली layer is going to glide even more. So what I'm seeing is at the periphery, I'm going to have a decrease in the velocity, and the maximum velocity is going to be in the center, and that's why it's going to form a parabolic curve, and this is known as a laminar flow. Okay? So the lamina which is in contact with the vessel wall has zero velocity. It might even have, that's known as a zero slip condition. And as we go towards the center, there's going to be an increase in the velocity. Now we have maximum amount of velocity that's going to be in the center. So this is known as your laminar flow. Okay? But just remember, यूजुअली अकॉर्डिंग, अगर मैं लैमिनर फ्लो की अगर मैं बात करूं, यूजुअली माई फ्लो इंक्रीजेज वेन एवर द प्रेशर इंक्रीजेज. इसका मतलब क्या है, हमारा लैमिनर फ्लो है, मतलब द प्रेशर इज डायरेक्टली प्रोपोर्शनल टू योर, मतलब योर फ्लो इज डायरेक्टली प्रोपोर्शनल टू योर चेंज इन योर प्रेशर. जितना प्रेशर चेंज इंक्रीज होएगा उतना हमारा फ्लो इंक्रीज होएगा. दैट इज अकॉर्डिंग टू लैमिनर फ्लो. बट व्हाट्स गोइंग टू हैपन इज, whenever pressure is going to exceed some amount, ठीक है, it's going to exceed some amount of number, or some amount of pressure is going to be exceeded, what are we going to have is this laminar flow now gets converted to your turbulent flow. And now I'm not going to see a linear rise in your flow as a unit of your pressure. Okay, and how do I calculate this, and what is this number known as? This number is known as the Reynolds number. It does not have any units. Okay, so this Reynolds number, whenever this Reynolds number is more than 2000, I'm going to see a change from the laminar flow, which was earlier, to a much less efficient turbulent flow. Okay, and let me just tell you one thing very clearly. हमारी बॉडी में there are very few examples of a turbulent flow. Usually हमारी सारी बॉडी में, whether it is pulsatile flow, I know कि हमारा जो arteries है they mainly flow in pulses, but whatever be the case, it's always a laminar flow and not a turbulent flow. We see turbulent flow in abnormal conditions like your murmurs. ठीक है, so just remember that. ठीक है, and just one more thing that we like to discuss is, whenever I'm going to have this, क्या होगा, मेरे RBCs are going to be at the center and I'm going to see plasma to be at the periphery, and this layer of plasma is going to be the marginal plasma layer. ठीक है, तो marginal plasma layer इस यहाँ पे होगी, and I'm going to see RBCs traveling in the center of that vessel. ठीक है, right. So what is this Reynolds number? So let's just calculate it. Reynolds number का जो main वो है, that is velocity times the diameter of that vessel, that velocity that is through which it is flowing, times the diameter of that vessel into your density divided by your viscosity. ठीक है, VdP by eta, where P is the density of that thing. ठीक है, you can use other units as well, but D हमने diameter के लिए use कर लिया, इस करके P is used as your density. ठीक है, all of these factors, whether it be the velocity or your diameter or your density or your viscosity, are going to affect your fluid momentum. And if they're going to affect your fluid momentum, they're going to affect your Reynolds number. And whenever the Reynolds number is less than 2000, I'm going to have a laminar flow. Whenever it is more than that, it's going to be a turbulent flow. Okay, and just remember, turbulence occurs normally in the ventricles. Okay, and can also occur in the human aorta during peak flow. उस time पे it might generate a murmur, which might be an innocent ejection systolic murmur. Okay, right. For example, in case of anemia, I often say that turbulent flowing हो सकती है. Why that can happen is because anemia में this viscosity is going to drop down. So whenever this viscosity is going to drop down, the Reynolds number is going to increase, and that's why I'm going to see a pulmonary ejection systolic murmur in case of anemia. That's known as a Hemmex murmur. Same can be said for pregnancy as well. Pregnancy, maybe there is a state of hemodilution, the hematocrit is decreasing, and that is the reason why I might see a systolic murmur in pregnancy as well. Okay? And whenever I'm going to see an increase in the velocity of your flow, that is via exercise, I'm again going to see an increase in the Reynolds number that might lead to an innocent murmur as well. Okay? All right. So this is about the Reynolds number and all of this. ठीक है, you already measured the blood flow measurement, हमने बहुत अच्छे से स्टडी कर लिया है, how we can use the fixed principle to measure that. ठीक है, limb blood flow हम measure करते हैं via plethysmography. ठीक है, and sometimes microvascular blood flow, if we have to measure, we measure it via the Ketty's tissue clearance using this radioactive substance known as xenon 133 and krypton. ठीक है, उसके through we can calculate the microvascular blood flow again on the principles of your fixed principle. ठीक है, all right. All right. ठीक है, so अभी we are going to discuss about the aorta now and its उसका waveform. So let's see that. ठीक है, so this is the aortic waveform there. All right. So just remember, when we calculate the mean pressures, a very important thing is when I need to calculate the mean blood pressure in the aorta, you know what is the formula? It's not the actual formula that we use to calculate. It's actually systolic blood pressure divided by diastolic blood pressure, systolic blood pressure plus diastolic blood pressure divided by 2. ठीक है, सो दिस इज इन द प्रॉक्सिमल एओर्टा इन द सेंट्रल आर्टरीज. बट जो हम यूजुअली ब्लड प्रेशर कैलकुलेट करते हैं फ्रॉम द ब्रेकियल आर्टरी, एंड वी से दैट इज योर डायस्टोलिक ब्लड प्रेशर प्लस 1/3 ऑफ योर पल्स प्रेशर. ठीक है, यूजुअली व्हाट हैपेंस इज at the level of the aorta, the diastolic blood pressure and aortic blood pressure usually have a similar role. They have their similar, उनका जो importance है वो similar है, इस करके we take it as the mean. But in case of peripheral arteries, because the diastolic blood pressure plays a very important role और उसकी waveform ज़्यादा है, इस करके we take as diastolic blood pressure plus 1/3 of your pulse pressure. Okay, just remember that. Now, what I'm going to see in this waveform here is This is a peripheral artery का waveform. So let's just see इसके waveforms के क्या क्या portions हैं. What I'm going to see is I'm going to see a forward wave. What is this forward wave due to? This is due to your stroke volume. The myocardium ने contract किया, ventricle contractility के कारण I'm going to see an increase in the pressure. ठीक है? Do you see this increase further? ठीक है? So this is known as your augmentation pressure. ठीक है? So I'm going to see an increase in the forward wave. That might be due to the increased contractility of the ventricle. But why am I seeing an augmentation pressure? And why am I seeing this wave, which is known as the reflected wave? This is known as a forward wave. This is known as a reflected wave. So the principle is very easy. What happens is, as हम aorta में या फिर किसी artery में we are pumping in blood, that will contribute to a forward wave. But what's happening is at the level of the arterioles or at the level of your vascular resistance, all of that blood flow doesn't want to go in these arteries due to resistance, and all of that blood might get reflected as a reflected wave, and that reflected wave might lead to augmentation in the pressure. Okay, so that is why I'm seeing a forward wave, and I might see an augmented wave as well. Why do I see an incisura and a dicrotic notch? That is due to the closure of the aortic valve. I might see an incisura and the dicrotic notch. Okay, I hope that's clear. And I've already discussed pulse pressure. We know it's systolic blood pressure minus your diastolic blood pressure, and it depends upon your stroke volume and it depends upon your arterial stiffness as well, the pulse pressure. ठीक है? So just see, ये मैं पहले भी discuss कर चुका हूँ that जो हमारे normal velocity of a blood flow है, you know how much that is. That is 20 centimeters per second. But it can only travel about 0.2 meters every second. But the pulse transmission velocity is about 4 से लेके 15 meters per second. Okay, and pulse it happens much faster at a much higher pace in your elderly. By a mean pace, I mean the transmission velocity of the pulse and not the actual rate. Actual rate is कम, bradycardia होता है ना older people में. But mainly the pulse transmission velocity is also dependent upon what this the studied the दूसरा factor was your arterial stiffness. तो जितनी हमारी stiff vessels होंगी उतना ज़्यादा pulse transmission velocity is going to increase. And because elderly में we have arteriosclerosis and increase in wall thickness, we're going to see an increase in the pulse transmission velocity. The pulse transmission velocity is very high and the blood velocity is very low. Okay, just remember that कि pulse पहले आती है and then the blood comes. मतलब whenever I want to accommodate this amount of blood that is there, I need to dilate the vessels that are present आगे. And what is this expansion known as? This is known as the pulse. So it's like we are basically receiving that amount of blood और उसके लिए हम preparations कर रहे हैं via this forward transmitting wave in the form of an expansion that's known as a pulse. And whenever the elasticity के कारण when it's going to recoil, so this expansion as well as a recoil which happens in arteries which we measure and which we measure via the radial artery or your carotid artery is known as your pulse. कि expansion भी है और recoil भी है. ठीक है? And the transmission velocity I've told you it's very varying 4 से लेके 15 meters per second. ठीक है? All right. So see one more thing. अभी हमने देखा था कंट्रोल में when we were seeing हमने देखा था कि जो हमारा एक हमारी मेन फॉरवर्ड वेव थी और एक हमारी रिफ्लेक्टेड वेव थी. यूजुअली दिस रिफ्लेक्टेड वेव इज वेरी इन द फॉर्म ऑफ अ डायस्टोली. ठीक है, दिस रिफ्लेक्टेड वेव इन केस ऑफ एन एओर्टा अकर्स इन डायस्टोल. ठीक है, इन केस ऑफ एन एओर्टा दिस अकर्स इन डायस्टोल. मतलब थोड़ा टाइम लगता है इसे. But whenever I'm going to have an increase in the systemic vascular resistance in case of mainly your hypertension, what's going to happen is instead of an increase in the diastolic blood pressure or your diastolic pulse waveform, I'm going to see a systolic inflection. मतलब systolic ही मेरी forward wave increase हो जाएगी. ठीक है? Now this is very deadly actually. Do you know why in mainly ischemic heart disease, why are we so scared of this hypertension? ठीक है? Mainly the thing is we needed a diastolic wave. हमें बहुत desperately चाहिए थी diastolic wave. Do you know why? Mainly because diastole is the time जहाँ पे हमारा ज़्यादातर myocardium is getting perfused via the coronary blood flow जो होता है ज़्यादातर, especially the subendocardial layers occurs during diastole. अभी हम क्या कह रहे हैं, जो हमें augmentation मिल रही थी during diastole हमने वो भी deprive कर दी है सिर्फ इतने चलते because we have not treated the hypertension. And what is that leading to? That leading to a systolic inflection और जो मेरी diastole में increase होनी चाहिए थी pulse waveform वो नहीं increase हो पा रही है. That is bad for us. ठीक है? So just remember in older individuals and hypertensive, the systolic pressure augmentation होगा systole में. Okay, and that's going to increase the cardiac work, that's going to increase the oxygen demand, as well as what will it do? It will also be depriving the diastolic increase that we were seeing. जो diastolic हमारे augmentation थी. ठीक है? And how we can prevent that? We can give GTN. ठीक है, ग्लिसरल ट्राइनाइट्रेट दो, आर्टीरियल कंप्लायंस इंक्रीज करो, सिस्टोलिक ऑगमेंटेशन में अपने आप कम कर दोगे वाया वेव रिफ्लेक्शन. ठीक है, right. We've already seen this. And as the peripheral wave, when it travels from the aorta to the peripheral artery, we're going to see some changes. What are these changes? First, systolic pressure wave increase करेगी. The incisura which we were seeing, incisura जो हम देख रहे थे, that's going to disappear now. Okay? Because incisura was occurring, your dicrotic notch, why was it occurring? It was mainly occurring due to the closure of the aortic valve, because we have transferred very far away from the aorta. As जैसे जैसे हम peripheral arteries की तरफ जाएंगे, हमारी dicrotic जो notch है, that's going or the incisura is going to disappear now. ठीक है? And the last में we're going to see a dicrotic wave. And what is a dicrotic wave due to? This is your reflected wave. Did I not tell you guys कि वो ज़्यादा reflected wave और ज़्यादा prolonged होएगी? So now this is occurring. That's your reflected wave from the periphery. That's a marker of your peripheral vascular resistance. ठीक है? All right. ठीक है. And what are the factors which affect your blood pressure? An age, increase in age will increase your blood pressure, especially your diastolic blood pressure as a result of your arteriosclerosis. Blood pressure is going to decrease during sleep. It's going to increase during your exercise, as well as just don't forget the gravity can influence on your blood pressure, as we know how the potential energy is going to fluctuate. Standing pay, the blood pressure might modulate, it might decrease for a while. Emotion and stress might increase, and your breathing, very important. दो waves हैं. We have something called the Traub Herring waves. Did I not tell you guys कि inspiration में हमारा heart rate increase होता है ना? And मैंने वहीं पे I told you a very important note was inspiration में हमारा blood pressure decrease होता है. ठीक है? So this fluctuations in your blood pressure which occurs during inspiration and expiration are known as your Traub Herring waves. ठीक है? Very important. ठीक है? So this Traub Herring waves, as you can see, inspiration and expiration, जैसे हमारा ventilation increase हो रहा है, देखो blood pressure कम हो गया. Then increase होएगा during expiration, then decrease होएगा during inspiration. So these waves are known as Traub Herring waves. ठीक है? इसी के very similarly, we have other type of waves that are known as Meyer waves. But these Meyer waves का actual mechanism we don't know. ठीक है? So these are these oscillations which occur in your blood pressure at a rate of 6 per minute. Occur at 6 per minute, मतलब 0.1 hertz. Okay? And these oscillations, they occur due to a random stuff. जो अपने आप जो peripheral arterioles है, वे vasodilate करते हैं, vasoconstrict करती हैं, and we are going to see these waves. They're known as Traub Herring Meyer waves. But if I have to tick one, I'm going to go with your Meyer waves, because Traub Herring waves हमारी inspiration के साथ fluctuations in your blood pressure. ठीक है? And what about your Valsalva? How is going to Valsalva कैसे affect करेगा हमारे blood pressure? पहली phase में what's going to happen is whenever the blood pressure is going to increase, whenever I'm going to perform Valsalva, पहले मेरा blood pressure is going to increase. Why? Because I'm going to have an increase in the intrathoracic pressure that's going to compress the thoracic aorta, that's going to generate more amount of blood pressure. That's one. In phase 2, because मैं Valsalva perform कर रहा हूँ, venous return is getting hampered, I'm going to see a decrease in the blood pressure due to decrease in the venous return. Once I stop the Valsalva, now what's going to happen? पहली चीज that's going to happen है जो thoracic aorta के ऊपर compression है, that's going to get relieved, and now the blood pressure will fall for a small amount of time. And now then उसके बाद, because the venous return gets augmented, I'm going to see an increase in the blood pressure. And last में, as a concluding response, I'm going to see increase in blood pressure के कारण a reflex transient bradycardia. So some people in clinics, in advanced clinics, I would say, they use this mechanisms, this Valsalva के fluctuations as a marker of your baroreceptor activity and your autonomic nervous system की intactness. मतलब अगर Valsalva के बाद आपको reflex transient bradycardia आया है, you don't suffer from autonomic neuropathy. ठीक है? Just remember the 4 phases, and I have depicted this as well. ठीक है? All right. Resistance, I already told you guys, according to Poiseuille's law, this is the resistance का formula. That is 8 eta L divided by pi R4. ठीक है? What is the eta? Eta is the viscosity. What is the L? L is the length, and R is the radius. And I told you resistance is inversely proportional to the R quadrupled. Okay? And that was the reason why whenever the radius is going to decrease by a factor of 2, I'm going to see an increase in the resistance by 16 times. Okay? Whenever it's going to get halved, I'm going to see an increase in the resistance by 16 times. Okay? Because the capillaries have the smallest amount of radius, we might be expecting यार इनमें तो problem नहीं हो जाएगी, इसमें तो resistance ज़्यादा होना चाहिए. But mind you, these capillaries are in mainly parallel. They are in parallel circulation. इसके कारण we are going to add via this. We are going to add their inversely proportionately. ठीक है? तो parallel arrangement के कारण जो net effective resistance in the capillary is going to decrease. ठीक है? The second factor which is going to contribute is capillary have a very short amount of length. तो उसके कारण resistance is further going to decrease. And just remember, capillary have this phenomenon known as your blood bolus flow, जिसके through they're going to decrease their viscosity. ठीक है? उसके through they can also contribute to a decrease in the resistance that is seen in the capillaries. We have already seen the Laplace law, but just remember, जब हम Laplace law blood vessels के बारे में बात करेंगे, now थोड़ा सा इसमें change ये हुआ है that the tension is equal to PR इज दिस इज फॉर द सर्कल, ठीक है, बट यहाँ पे हमारा जो लैपलेस लॉ है दैट इज टी इज इक्वल टू पी आर, ठीक है, और वी कैन से पी इज इक्वल टू टी डिवाइडेड बाय आर, ठीक है। वहाँ पे इट वाज व्हेन वी वर डिस्कसिंग द हार्ट इट वाज द टू वाला फैक्टर वाज आल्सो देयर, ठीक है, सो दिस इज योर लैपलेस लॉ, ठीक है, ऑलराइट, ठीक है। All right. Actually, circulation, I think we might not be able to cover. The last topic जो मैंने I thought of discussing with you guys was regarding the cerebral circulation and your coronary circulation. But I guess we can cover coronary circulation when we might discuss your cardiovascular pathology, and we might discuss your cerebral circulation when we discuss your CNS. ठीक है? तो इस केस में we are mainly done with your CVS physiology. If you look at Gnong के सारे chapters I've tried to cover them all. And all of these units, all of these stuff, that is, one chapter is on cardiac output, one chapter is on cardiovascular regulatory mechanisms, one chapter is on electrophysiology of the heart. Okay, all of these chapters have been covered. One special chapter was on your special circulations, जिसमें हमारा coronary and your cerebral circulations पे ही I see maximum amount of questions coming. ठीक है, so we can do that, or you can refer to my notes as well, and you can study that on your own for the time being. And I think आज का session was good. I think we covered a lot in about 1 hour and 20 minutes. And considering the 10 minutes wait time as well, I think that's enough. And I'm going to share all of these slides at the Telegram group so you can look at it. So it's about 220 pages. And its main thing is pura CVS physiology has been covered over here. Okay. I hope you like this lecture. And please give me a thumbs up. Please share it with your friends. अगर कोई आपको question है, you can ask it. ठीक है, you can ask it अभी, and I can reply. And again nahi hai, then we can further plan, and I'll see you guys tomorrow for some other portion. With this, almost my physiology has been covered. One unit that is left is your respiratory physiology that I might take up kal se, and then we might be done. Thank you Shmangi, thank you for conducting this session. So aaj ka basically my session has been productive. I hope you go back and just read these chapters from Gnong as well. Okay. All right, so I'm going to end this session now. Thank you all for joining.